All right, everybody, welcome back to part 2 of the nervous tissue lecture. In this part, we're going to talk about the physiology of nervous tissue. So we're going to talk about the relationship between current, voltage, and resistance. We're going to talk about some different ion channels, talk about something called resting membrane potential, graded potentials, and action potentials. We're going to talk about how these action potentials are generated and discuss refractory period and solitary conduction. And then finally, we're going to discuss the synapse as being electrical and then being chemical. And finally, we're going to end with a discussion on neurotransmitters. So with that being said, let's go ahead and get started. As a general reminder, the nervous system is the master controlling and communicating system of our body. Okay, so I brought this up in the part 1. I just wanted to remind you, in case you forgot, this is our master control and communication center. And the human body as a whole is what is called electrically neutral. So in its entirety, the human body has the same number of positive and negative charges, which is why humans in general can't conduct electricity, right? We are electrically neutral beings. There are regions, however, within the human body that are more positively or more negatively charged. And because, for example, these opposite charges are attracted to one another, we have to use energy in order to keep them separated. So I'm going to give you the example here. We'll start out with this picture over here, where I just created, we're going to say this is a cell inside of here, and here's the nucleus of the cell. And we have 4 negative charges inside of this cell, and there's no positive charges inside of this cell. So overall, we would call this cell being a negative net charge, having a negative net charge. But say just outside of the cell, if this blue line is the membrane, say just outside of the cell, we have 4 positive charges, all right? And so this would be in maybe the extracellular fluid, okay? And that's the fluid that bathes our cells and keeps them nutrient healthy, nutrient rich, right? So say outside of this cell, we have 4 positive charges. Okay, so we're going to say then there's no negative charges out here. So the net charge out in this space, in our extracellular fluid, we're going to say our net charge here is positive, right? So because these 2 charges, these charges on the inside being negative and these charges on the outside being positive, because they're attracted to each other, it actually takes energy to keep them separated, right? So kind of going along with that, then if 2 opposite charges, so if this positive and this negative come together, then we create energy, and that energy can be used to do work. So because of the way that this is set up, keeping opposite charges separated creates what we call potential energy. So basically, potential energy is just energy that could be created if these charges came together, but since they haven't come together, that energy is just potential. It could potentially be created, which is why we call it energy created by keeping these charges separate. That's why we call it potential energy. Okay? So I'm gonna try to explain this in the simplest terms that I can, and I'm gonna warn you, it's a little bit physicsy, but it's so important to understand, and I'm gonna try and break it down to make it really easy for you guys to understand. So we just said that keeping charges separated creates potential energy, right? So this situation, keeping these charges separated, is creating potential energy. okay? And we can actually measure how much potential energy is being created here. And we call the value of the potential energy, okay, so like the number associated with how much potential energy is here, we call that voltage. Okay, so voltage is just a measurement of potential energy. So in cells, like we have a cell here, the charge difference that's created across this plasma membrane results in what we call a membrane potential. And basically, this is just the technical term for saying the potential energy that's created across this membrane, okay, because these charges are keeping separate or being separated. And in cells, we actually always measure the charge on the inside of a cell relative to the charges on the outside. So if a cell has more negative charges inside than what there is outside, we say the cell has a negative membrane potential, like in this example, right? So in this example, we have 4 negative charges on the inside of the cell. We don't have any negative charges out here. So compared to the outside, the inside of the cell is negative. Okay, this side of the cell membrane is negative. Alternatively, if these were flipped around and these are all positive charges and there was no positive charges out here, we would say that the membrane potential is positive. The cell has a positive membrane potential if all the positives were in here and there were no positives out here. So this membrane potential, aka potential energy created across the cell membrane, can actually be measured using what's called a voltmeter. And then the voltmeter, as you can kind of infer, hopefully, is going to measure how much potential energy there is across this cell membrane. And again, the numerical value that the voltmeter gives us is called voltage. So I just like threw some numbers in here. This is completely made up. I just did it for simplicity's sakes. If we stuck 2 electrodes, one inside of the cell, one outside of the cell, and we looked at the voltmeter, it would give us some negative number here because of the way that this is oriented, okay? Now, the next point I want to make is that the greater that the charge difference is between 2 points, the higher the voltage will be, okay? So what I mean by that is, let's look at this top picture first, all right? And in this top picture, we have 2 positive charges on the outside, 2 positive charges on the inside, but 4 negative charges on the inside. Well, we can cancel out the charges that we have here. These 2 can be canceled because they balance each other out, okay? So we're going to actually just pretend like those aren't there, all right? And so in this case, if we place an electrode here and here, then we would see that the charge difference here inside the net charge is -2, okay? And we're going to say that the inside of this cell has a -2 net charge, versus this bottom cell down here where we have 4 negative charges, so -4. Because of this is farther away, this cell down here, it's farther away from 0 than the top cell. This is going to be have a higher voltage, okay? Just because this number is farther away, if we did, I don't know, take a number scale here, right? This is 0, here's -2, here's -4. -4 is farther away from 0 than what -2 is. And this will result in us being given that this cell down here has the higher voltage, okay? So I just wanted to point that out before we move on here. Okay, so now that we have a general understanding of voltage and potential energy, let's take that information and apply it to neurons, because we are talking about nervous tissue here, right? So the situation that we just described on the last few slides can be related to neurons. So at rest, a neuron has a voltage of negative 70 millivolts. Okay, so a resting membrane potential of minus 70. All right, so this is really similar to the situation that I just had created that little animation sort of on the last couple of slides. We're looking at the cell, at the neuron, and we measure the charge inside relative to the charge outside. The inside is much more negative than what the outside is. So the resting membrane potential here of a neuron is negative. So in this case, it's actually negative 70. The cytoplasmic side, again, is the cytoplasmic, that'd be the inside, is negatively charged relative to the outside. So even if there were negative charges floating around out here, there's still a lot more on the inside. So we still say that it's negative relative to the outside. Okay, so the actual voltage of a resting neuron, it's quite variable. It could be between minus 40 millivolts to minus 90 millivolts, but as a general rule, and for testing purposes, our resting potential is minus 70 millivolts. So the question that you should be asking, and if you're not, then maybe I'm going to stimulate you to think about, why is the inside of this cell negative compared to the outside? What properties about it make it negative compared to the extracellular environment? And over the next couple of slides, we'll discuss this. So we're going to talk about these reasons, including the differences in concentrations of ions of the intracellular fluid and extracellular fluid, and then also this negative membrane potential is due to the difference in plasma membrane permeability. All right, so we're gonna really focus on this over the next couple of slides. Okay, so we're going to start here with how we even create this situation where the inside of the cell is negative. Okay, so how are we creating this resting membrane potential? And remember, I said one of the reasons is because there's differences in the amount of ions inside relative to outside. Okay, so let's consider this. Sodium molecules have a higher concentration, so there's more sodium molecules outside of the cell than there are inside of the cell. Okay? Alternatively, we have more potassium ions inside of the cell compared with outside of the cell. Okay? It's important to know that potassium is going to be playing the most important role in establishing this membrane potential. So I have already then covered one of the reasons why the resting membrane potential is negative relative to the outside here. So the other reason is because of these differences in membrane permeability. So let's kind of talk about this, and we're going to go through this little sort of scenario over here in this panel, and we're going to build essentially our resting membrane potential step by step. Okay, so let's pretend for all intents and purposes that a cell only has potassium channels, and potassium channels, potassium itself as an ion, it's 25 times more permeable than what sodium is, and it diffuses out of the cell because remember, there's already more potassium inside of the cell, and so it's going to diffuse down its concentration gradient, go from that crowded gym to the hallway space. So it's going to move from inside to outside, okay? So potassium is diffusing out of the cell. So if a cell only has potassium channels and potassium is diffusing out of the cell, what's going to happen? Well, since potassium has a positive charge and we have a positive charge leaving the cell, that is going to result, oops, I'm sorry about that. Let me go back. Okay, since we have a positive charge leaving the cell, we have this positive charge, it's leaving, that's going to leave us negative on the inside, right? Because if you take away positives, you can only take away so many positives before you reach negative, right? So let's put that on a number line, so just so we can envision this happening. Okay, if here's 0, and this is our positive side, and this is our negative side. If you're taking away positives, eventually you're going to cross over into this negative territory, right? So that's essentially what's happening here in this first panel. So if we only had potassium channels and potassium's leaving the cell, we are going to leave the cell with a net negative charge inside, okay? Now let's add in some sodium channels. And sodium is it's only slightly permeable across the plasma membrane. And because we started out in a scenario where sodium is really high on the outside of the cell and really low inside, sodium diffuses down its concentration gradient and moves in to a cell, okay? So sodium is going to come in. But like I mentioned before, potassium is 25 times more permeable than what sodium is. So because of that, we're maybe going to have 2 potassiums leaving. So 2 positive charges leaving, but only 1 positive charge coming in. Okay, so on our number line, if we start over here and we have 2 positive charges leave, and then we only have 1 come back in, and then maybe 2 more leave, and only 1 comes back in, you can see that we're losing more positive than we are gaining positives. And so this alone is going to lead to us having a negative charge on the inside of the cell, okay? So more potassium is diffusing out than sodium is coming in, and so as a result, the inside of the cell is more negative, or the other way you can think about it is it's less positive than what it would be otherwise. And so this is actually helping to establish our resting membrane potential. Okay, and there's one more thing that I would like to point out here is that we have these things called sodium potassium pumps. Okay, and this should not be anything new to you because we talked about it. It's been a while, but we have talked about it before in class. So our sodium potassium pump actually helps to keep this resting membrane potential stable. Because like I talked about in this second panel, we're going to have 2 loss positive charges, 1 gain, 2 loss, 1 gain. And if we kept at this sort of pace, the inside of the cell would keep becoming more and more and more and more negative over time. Okay, so but we don't want that. We want our cells to have a membrane potential that is relatively constant. Okay, so it doesn't go all haywire. And the way that we do this is via the sodium potassium pump. And the sodium potassium pump is going to maintain our gradients for potassium and for sodium because it is going to pump out 3 sodiums and bring 2 potassium molecules in. Okay, so let's think about what that's doing. If we have 2 potassium ions leaving, but we're bringing 2 back in. Great, that's awesome. We are canceling each other out because what we lost, we just gained right back. Alternatively, we're bringing 1 positive in for every 3 out. This is going to then lead to keeping this negative charge here. So a cell at rest is it's kind of like a leaky boat. So potassium is leaking out and sodium is leaking in, but our bailing pump, basically the water pump here for this boat, is like the sodium potassium pump, which is going to transport sodium back out where it belongs and bring potassium back in where it belongs. But let's consider one more thing, all right, before we move on. Remember up here how I said that sodium is really high outside the cell and really low inside, and potassium is really low outside, and sodium and potassium is really high inside? Okay, let's look at the direction that the sodium potassium pump is pumping things. Normally, if we're talking diffusion, things are going to flow down their concentration gradients, right? So from high to low. This is a diffusion type situation, right? However, if we notice with the sodium potassium pump, we are moving sodium against its concentration gradient. We're pushing people into a crowded gym that don't necessarily want to go in there, but we're pushing them anyways. Okay? To do this, and essentially you can see we're doing the exact same thing with potassium. We're moving it against its concentration gradient. In order to accomplish this, in order to move things against their concentration gradients, this costs energy. Okay, so and in the case of the sodium potassium pump, it costs 1 ATP molecule every single time that we pump in 3 potassiums and pump out 3 sodiums using this pump. Okay, so just bear that in mind that keeping this resting membrane potential does have a cost, and that cost is energy. And this relates back to our first slide here, this one, how we say that keeping opposite charges, here we go, because opposite charges are attracted to one another, you have to use energy to keep them separated. That energy in our case is ATP. okay? ATP is keeping these charges separated, keeping this resting membrane potential at minus 70 millivolts, okay? So we have learned this stuff before, but I just want to give you a quick reminder as to how ion channels work. Since we just talked about some channels specifically, we talked about the leaking potassium and sodium channels, and then the sodium potassium ATPase. But quick reminder of how ion channels work. We have three main types of gated ion channels. So the channels that I discussed on the last slide, go back one here, these are not gated. Okay, these are just leakage channels. Right, so this is for diffusion. These are just diffusion channels. There's no gates that are blocking potassium or sodium from entering or leaving. Okay, those are non-gated. But we have gated channels, and the first type that I will talk to you about is, or remind you, I guess, about, are the chemically or ligand gated channels. And these are the channels that open in response to the binding of some kind of chemical on a receptor, for example, like a neurotransmitter. So let's pretend like this is a sodium potassium channel, a sodium potassium gated channel. And when the receptor site is not bound, the channel is closed. Potassium can't leave, sodium cannot come in. The gates are closed. However, when a neurotransmitter binds to its receptor site on this channel, then the gates open and sodium is free to come in and potassium is free to leave. Okay, so that's just a reminder of chemically gated channels. Voltage gated ion channels are going to open and close in response to changes in that thing called membrane potential that we were talking about. So in the first case here, we see a voltage gated sodium channel. And normally when we have negative charged membrane potential, our gate is closed. Okay, so as you can see there, we're closed. All right, and that means it means sodium cannot make it through. All right. However, if a situation occurs where the inside of the cell now becomes positive, that is going to cause the gates on this channel to open. And now sodium can enter the cell. Okay, so voltage gated channels open and close in response to changes in our membrane potential. Okay. The final type are the mechanically gated channels. And these ones are a little bit more rare, and we don't really talk about them very much, but mechanically gated channels only open and close in response to a physical structural change of the receptor. So think about your sensory receptors for touch or for pressure, for vibration. In a normal situation, before we have any change, the channel is closed, okay? So ions can't move in or out of the cell. However, if you touch something with your fingertip, the receptors that are bound in your fingertips to each cell membrane, these receptors are being physically moved because your finger just pressed up against something. They're physically changing their shape. And that will allow ions to move into the cell or out of the cell. And once the pressure is removed, once you lift your finger back up off of the table or whatever you're pressing down on, then the gate will close again. Okay, so I just wanted to remind you guys of how these ion channels work. So now we're going to watch a short video on resting membrane potential. And this does a really nice job sort of summarizing what we were just talking about. Okay.
The human brain alone contains about 100 billion nerve cells called neurons. A neuron, like all cells, has negatively and positively charged ions in the fluids located both inside and outside of the cell. However, the ionic composition of these two fluids is markedly different. The inner surface of the membrane is negatively charged, while the outer surface is positively charged. This unequal distribution of charge creates a voltage across the plasma membrane, known as the membrane potential. In a resting, unstimulated neuron, this membrane potential is known specifically as the resting membrane potential. The value of the resting membrane potential depends on two factors. The first is the presence of concentration gradients across the plasma membrane, especially those gradients for sodium and potassium ions. The second is the permeability of the plasma membrane to sodium and potassium. In the resting state, this permeability depends on the number of non-gated leak channels present in the membrane for these ions. The magnitude of the resting membrane potential, inside relative to outside, is measured in millivolts using a voltmeter and 2 electrodes. We'll now place both electrodes in the extracellular fluid. Note that the voltmeter records 0 voltage. This is because the unequal distribution of charge exists only across the plasma membrane. The intra- and extracellular fluids are otherwise electrically neutral. However, when we insert one of the electrodes into the intracellular fluid, a voltage is recorded. The value of the resting membrane potential varies, but on average it is minus 70 millivolts.
So I think this is a really good video summarizing resting membrane potential. So hopefully you found that to be helpful in its 3D world there. Again, as a reminder, if you need a few more seconds to answer the question, I highly encourage you anytime you see a clicker question to just pause the video, read it, answer it, and then resume the video, and the answer will come up within a couple of seconds. So in the PowerPoints, I have these, after giving you the answer, this question came from this slide. You can find the information directly from this slide. So now that we know what a resting membrane potential is, we need to understand how we can change that resting membrane potential, because changes in the membrane potential are used as signals to receive, integrate, and send information around the body. So this is functionally how neurons are transmitting information around the body. So membrane potential changes when one of two things happens. One, the concentration of the ions across the membranes need to change, and or two, the membrane permeability has to change. So channels need to open or close. And the changes, these changes, will produce two types of signals. One, they'll produce graded potentials, And graded potentials are just incoming signals that operate over really short distances. And or two, the changes will produce action potentials. And action potentials are the signals along the axons that travel really long distances to relay information. So let's talk about each of these things in more detail. So changing the resting membrane potential. The terms that we use to describe membrane potential changes are relative to the resting membrane potential. So by now we know that the resting membrane potential is minus 70 millivolts, and we call it as polarized. So resting membrane potential is polarized at minus 70 millivolts. Check. If we change the resting membrane potential and we depolarize the membrane, we are referring to a decrease in membrane potential, aka the membrane potential is going to move towards zero. The inside of the membrane is going to become less negative than what the resting membrane potential was. For example, now, if we have our resting membrane potential at minus 70 and we go through a depolarizing state, maybe it becomes minus 50 millivolts instead of minus 70. Depolarization as a whole increases the probability of producing an impulse. Okay. The other term that we'll use sometimes is called hyperpolarization. And hyperpolarization increases our membrane potential, so it moves it even further away from zero. The inside of the membrane now is going to become more negative than what it was at rest. So maybe now, instead of being minus 70 like it was at rest, we have a stimulus that causes the membrane potential to drop down to minus 90 millivolts. Hyperpolarization will decrease the probability of firing an impulse or an action potential. Okay, so the further away from zero you get, the harder it is for a neuron's membrane to fire an action potential. Okay. So I mentioned action potentials and graded potentials, but let's just talk for a second about graded potentials. Graded potentials are these really short-lived local changes in the membrane potential. It's important to know that the stronger the stimulus is, the more voltage is going to change, and the farther the current is going to flow. So graded potentials are stimulated or triggered, by a stimulus that's going to open the gated ion channels that are present in the membrane. This is going to result in depolarization or sometimes hyperpolarization. And graded potentials are named according to their location and their function. So before I talk about that for a second, let's just consider this. So we have some sort of stimulus come in and it's going to land right here on the plasma membrane. And the inside we know normally is negative, but since this is a depolarizing stimulus, that causes the inside of our cell to become positive, right? So a small patch of the membrane is going to depolarize. Because opposite charges attract each other, this is going to create local currents, which are represented by these black arrows, that are going to depolarize the membrane areas that are right next to the original stimulus. So we know the original stimulus was this one here. Well, because these opposite charges, it's sitting next to opposite charges, it's attracted to those opposite charges that are sitting right next to it. And so that's going to create a current. And that is going to spread the depolarization from this one spot to now maybe this whole section. But as you get further and further away from this original stimulus spot, the current starts to decay, right? So with distance, the membrane potential change is decaying because current is going to be lost through the leaky channels that are present in this membrane. Okay, and so consequently, this is the reason why graded potentials are really short-lived and relatively localized. Going back to their naming, receptor potentials, they're also called generator potentials. These are graded potentials that are going to be present in receptors of sensory neurons, okay? So they're called receptor potential because they are located in receptors. And then it's also sometimes referred to as a generator potential because it's in sensory neurons, which we know sensory neurons are going to then transmit information to the central nervous system, which will then send out a motor signal. The other type of graded potentials we have are postsynaptic potentials, and these are neuron graded potentials. Okay, and so we're going to talk a little bit more about that later. But as a whole, you can see that if the original stimulus is right in this section on the neuron, that section will be highly depolarized or move more towards zero. So if on this number line, we had 0 being maybe here, right? We can see that a depolarizing stimulus is going to move us towards 0. And that depolarizing scenario is going to occur in a very local place on the neuron. As you get further away from where that stimulus occurred along the neuron, the less change that occurs down there, okay? The next thing that we're going to talk about is action potentials. Action potentials, this is a very important concept to understand because this is the primary way that neurons are sending their signals. So action potentials are just a means of very long distance neural communication. It's important to know that action potentials only occur in muscle cells and in the axons of neurons. In action potentials, essentially what happens is we have this really brief reversal of membrane potential with a change in voltage of like 100 millivolts, give or take. And it's also important to know that unlike graded potentials, action potentials do not decay over distance. So in neurons, action potentials are also sometimes called nerve impulses. And action potentials then involve the opening of certain voltage-gated channels, okay? So key players in action potentials are going to be sodium channels and potassium channels, voltage-gated sodium channels, and voltage-gated potassium channels. So voltage-gated sodium channels, they have 2 gates and they alternate between 3 different states. All right, so what do I mean by that? 2 gates. Here's a gate. There's a gate. All right, we have 2 gates. At rest, what we see here at rest, both of the gates are, I'm sorry, the gate is closed, okay? So no sodium can enter the cell. During depolarization, this gate that used to be present here is now open. sodium can now enter. And then we have an inactivation state. So after a certain time period has lapsed, this gate that used to be sitting down here like this swings over and blocks this voltage gated sodium channel. And now sodium again can no longer enter. Okay, so 2 gates in 3 different states. Potassium channels, voltage gated potassium channels, they only have 1 gate and they are either open or they're closed. So during rest, closed at rest. Potassium gate is, you see that gate right there, is closed. After depolarization occurs, there's a brief delay, and then potassium gate opens up so potassium can leave. Okay. All right, so 1 gate, 2 different states. So these are kind of our key players here for action potentials. And the big picture, what is this graph showing? So this graph is showing that during the course of an action potential, the voltage is going to change over time at a given point within the axon. So at resting state, our voltage is this minus 70 millivolts, and there's no ions that are moving. And then depolarization is going to occur, and that is when the sodium channel, the voltage gated sodium channel, is going to open and sodium is going to flow into the cell. And then we're going to reach a peak point where, again, after that brief delay that we talked about with potassium, voltage gated potassium channels, then we reach a point where the voltage gated potassium channels open. And this is approximately the point also where the inactivation gate of the sodium channel swings into place. And so now we have this point of repolarization. So potassium is flowing out of the cell. And then we hit our resting area again, but we dip a little bit further because potassium channels are kind of slow when compared with sodium channels. So the potassium channel, it hits that resting membrane potential, and then it's like, oh crap, I need to close. Okay, and so then the potassium channel does close, and then we get back up to our resting state due to the action of the sodium potassium pump. Okay, so this is big picture. So let's break this down a little bit further. All right, so how do we generate this action potential? There's 4 main steps. We're going to have our resting state, depolarization, repolarization, and hyperpolarization. So, resting state. In the resting state, all of the voltage-gated sodium and voltage-gated potassium channels are closed. Okay, so sodium can't come in, potassium can't leave. We have some leakage channels present. So let's pretend here like we have some leakage channels present. So this is a leakage channel, and then maybe we have a leakage channel over here, which is fine. So some potassium and sodium movement is occurring, but for the most part, we're maintaining our stable resting membrane potential during this resting state. So our membrane here is going to be charged at minus 70. So during resting state, we are at minus 70 millivolts. okay? And all the gates are closed, right? And then depolarization happens. Depolarizing local currents are going to open our voltage-gated sodium channels, and sodium is now going to rush into the cell, okay? And so let's consider that just for a second. We started out at minus 70 inside the cell, right? So we started out at minus 70, but now we have positive charge coming in. That means that we are going to move towards 0, right? Because now we're adding positive charges to this negative, okay? And so at this point, sodium is rushing in, and the rushing in of sodium is going to cause even more depolarization, which is going to open even more voltage-gated sodium channels. And so as a result of all of these sodium channels being opened and sodium rushing into the cell, the intracellular fluid becomes less negative, or it moves towards 0. We reach a point at this depolarization thing that's called threshold. And so once the membrane potential reaches minus 50 to minus 55 or so millivolts, that's when the positive feedback is causing the opening of all the sodium channels. So up here I said sodium influx is going to cause more depolarization, which opens more sodium channels. And once we reach this point, minus 50 to minus 55 millivolts, that's kind of when we're at a point where we're like, all right, let's go for it. Let's open all of the channels. Why not? Okay, so that's kind of where we're at with threshold. And because we decide to open up all of the sodium channels, that's going to result in this really large action potential spike, and the membrane polarity jumps all the way to 30 millivolts. Okay, so we started at minus 70, but by the end of depolarization, inside of our cells, oops, sorry about that. Go back for 1 second. The end of depolarization inside of our cell is now plus 30, okay? All right, so once we reach this plus 30 mark, then what happens? What happens next is what's called repolarization, okay? And in repolarization, the sodium channels are inactivating, and this is when the potassium channels are going to open. All right, so during repolarization, we have that inactivation gate swinging into its spot. And so at this point, the membrane is no longer permeable to sodium. Okay, so it's going to decrease permeability to the resting state. So we still have those leakage channels present, so sodium can still flow that way, but remember, sodium's not very permeable across this membrane. So most of our permeability comes from the opening and closing of this gate. And now that this inactivation gate is swung into place, sodium can no longer enter. Okay. However, what happens kind of simultaneously is as the inactivation gate of the sodium channel is swinging into place, the potassium channel's gates open. And now potassium can leave the cell. All right, so now we have a positive charge leaving the cell, which means that the membrane inside here, this part of the membrane, is going to start becoming negative again, right? Which is why we see our graph here moving back downwards, okay? So we're going back to normal. Okay. So just kind of to summarize this then, repolarization is going to reset our electrical conditions. Okay, it's not resetting our ionic conditions, it's resetting our electrical conditions. And after repolarization is over, that's when the sodium potassium pumps are going to restore the ionic conditions. So get the same number of ions inside and outside as what there was originally. Okay. So we have one more thing to talk about with this action potential, and that's called hyperpolarization. So remember how I said potassium channels are kind of slow? So we reach our resting state, and then potassium channels are like, oh crap, okay, it's time for me to close. But by the time they close, our our membrane potential, excuse me, is already down at maybe minus 90. At this point, this is when during hyperpolarization, this is the point of I should have closed already and I'm going to close. It's kind of that gray area in between those two. And so during hyperpolarization, we have some of these potassium channels remaining open. And at the same time, that's when the sodium channel is going to reset. So we see now the sodium channel is resetting. So we move the inactivation gate down out of its place. We closed our locking gate. And so now we're ready for the next action potential. But because some of these potassium channels are still open because they're slow, the inside of the membrane is going to become even more negative than what it is at resting state, and thus lead to what we call hyperpolarization, which is a dip below the resting state. And so just to kind of put that into perspective, here would be our resting state, this line. And you can see that we've dipped below that line because the potassium channels are slow at shutting and they accidentally let too many positive charges out. Okay. All right, so here is a nice little video summarizing everything we just talked about in generating an action potential.
Neurons communicate over long distances by generating and sending electrical signals called action potentials. Most action potentials originate in the axon's initial segment, a region also called the trigger zone. During an action potential, voltage-gated sodium and potassium channels open and close, thus altering the membrane's permeability to sodium and potassium ions. As these ions diffuse through the channels, the membrane potential changes. It depolarizes from the resting state of minus 70 millivolts to a peak value of plus 30 millivolts, and then quickly repolarizes back to minus 70 millivolts after a brief period of hyperpolarization. In this topic, we explore, step by step, the events that occur as an action potential is generated in the axon's initial segment.
Okay, so before we move on, I'm just going to talk about the thing that I mentioned earlier about threshold. So remember I said once we reach minus 55 to minus 50 millivolts, that's when all of the sodium channels are going to fly open, and that's when we have that huge spike in that depolarization wave. But it's important to understand that not all depolarization events produce action potentials. So for the axon to actually fire, the depolarization has to reach that threshold voltage to trigger the action potential. So we can depolarize just a little bit. So say if we had some kind of depolarizing stimulus and we started depolarizing, but we only got to there. If we only got to there, we haven't met our threshold value. Okay, so as you can see from the diagram here, if we only got to here, we are not at our threshold value, and therefore this would not lead to an action potential. So at threshold, the membrane has been depolarized by 15 to 20 millivolts, and this is going to lead to the sodium permeability increasing, and that sodium influx is going to cause potassium efflux, and then the positive feedback cycle is going to begin. And so at threshold, we reach this point of, yes, we're going for it, right? So it's all or none, and an action potential is either going to happen completely or it's not going to happen at all. Okay, so we have to change our membrane potential by at least 15 to 20 millivolts in order for an action potential to occur. So all of the events of the action potential that we just talked about, they only happen at one small section of a neuron at a time. Okay, and so this next sort of section here is on propagation of the action potential, and propagation allows the action potential to be transmitted from one section down the entire length of the axon towards the axon terminals. Once initiated, so once you've started the action potential at one section, then the action potential is going to self-propagate. It's going to go all on its own down the entire length of the axon, because sodium influx through voltage gates in one membrane area are going to cause local currents that cause the opening of sodium voltage gates in the area that's right next to that area, and then that's going to cause the depolarization of the next area. And so that's kind of what you're seeing along this sort of graph here. If we started out back here, all right, so if we start out with our depolarizing section right here. Right, so that's where the initial stimulus was. Okay. From here, what happens is the change in membrane potential that occurred in that section will cause the next section to change its membrane potential. And then the next section, and then the next section. So we see this membrane change occur down the length of the axon, okay? And so this is kind of how it's going to move along, all right? So let me just go back and start from the beginning, and I'll play this kind of quickly so you can see the change. So look, pay attention to this area right here as I click through the next couple slides. Right, so you can see as it's moving, how it's moving down the length of the action potential, or down the length of the axon, excuse me. All right, so let's talk, I'm going to show you this quick little video on propagation of action potentials.
Action potentials propagate in a continuous fashion in unmyelinated axons. Once an action potential is generated in the initial segment of the axon, it propagates the entire length of the axon. Recall that a threshold stimulus causes voltage-gated sodium channels to open. The influx of sodium ions generates an action potential. It also establishes a depolarizing current that flows to the next segment and brings it to threshold. Voltage-gated sodium channels open, regenerating the action potential in this segment of the axon. Current flows from this segment and depolarizes the next segment to threshold, thus regenerating the action potential yet again. In this way, regeneration continues in one direction all the way down to the axon terminals. The basis for unidirectional propagation is revealed when we take a closer look. By the end of the depolarization phase of the action potential, all voltage-gated sodium channels inactivate and voltage-gated potassium channels open. These two events render this segment of the axon temporarily insensitive or refractory to another depolarizing stimulus. However, voltage-gated sodium channels in the downstream segment are closed and receptive to a depolarizing stimulus. Thus, propagation occurs sequentially down the axon to the axon terminals. In myelinated axons, action potential propagation is a bit different. Here they propagate in a saltatory or leaping fashion. The myelin sheath consists of multiple layers of tightly wrapped glial cell membrane. But this sheath is not a continuous one. Exposed areas of axonal membrane, known as nodes of Ranvier, occur at discrete intervals. Voltage-gated sodium channels are abundant in the nodes, but largely absent between nodes. So, action potentials are regenerated at each node, not in areas covered by the myelin sheath. However, the myelin sheath does provide the insulation necessary for the rapid spread of depolarizing current. And the sooner the nodes reach threshold, the faster action potentials propagate along the axon. Saltatory conduction is extremely fast. Velocities often exceed 100 meters per second. In contrast, continuous conduction is fairly slow. Velocities rarely exceed 2 meters per second. Nevertheless, both continuous and saltatory conduction propagate action potentials over varying distances because action potentials regenerate along the way. Summary: propagation of an action potential. Once generated, the action potential propagates the entire length of the axon without decrement.
So now we're going to consider whether or not there are times when a neuron can't fire an action potential. And the answer to this question in short is yes. Action potentials have these things called refractory periods, or times when the neuron can't trigger another action potential. This occurs when voltage-gated sodium channels are already open, so the neuron can't respond to another stimulus. And we have two different types of refractory periods. We have the absolute refractory period and the relative refractory period. So the absolute refractory period is the time from the opening of the sodium channels until those channels reset. And so on our image over here, absolute refractory period is during this time frame. So easy. When the voltage gated sodium channels open until they reset. And the purpose of having this absolute refractory period is to ensure that each action potential is an all or none event. Okay, so this is actually what helps to enforce one-way transmission of nerve impulses. And what I mean by that is this helps to ensure that along the axon, that the action potential keeps moving one way. All right, so this would be the purpose of having an absolute refractory period. The other type of refractory period we have is what's called the relative refractory period. And during the relative refractory period, basically it coincides with the hyperpolarization state. And so let's remember that basically anything below 70 millivolts, we're going to be hyperpolarized. So relative refractory period is when most of the sodium channels, they've returned to their resting state, but some of the potassium channels are still open, and that's causing the membrane to repolarize still. And so in this period, the threshold for generating another action potential is going to be even higher. So the only thing that's going to actually cause another action potential to occur during this period would be a very, very strong stimulus. So I just want to briefly explain what I mean in this scenario about the threshold being higher. So remember that normal threshold is, we're going to put it about, okay, say it needs to be about right here. This needs to be our threshold. Okay, so we have to reach this point, give or take. But at resting membrane potential, we start out resting membrane potential, we start out here. Okay, so at rest, we only have to change 15 to 20 millivolts to have that all or nothing phenomenon. However, if we are in this hyperpolarized state, okay, so we're down here in this hyperpolarized state, in order to reach threshold, now we have to change this much. Okay, and so the only thing that's going to cause that big of a change would be a really, really strong stimulus. Okay? One way to kind of think about this, and this is something that the Mary textbook points out, is like you think refractory dog. If the dog is absolutely refractory, then he's never going to come to you when he's called. But if he's relatively refractory, maybe he'll come to you if you yell loud enough at him. Okay, and so that's kind of comparing absolute refractory to relative refractory in terms of this action potential. So if we are absolutely refractory here, then it doesn't matter how much stimulus you give, you can't fire another action potential. It's just not going to happen. But in the relative refractory period, if you yell loud enough or if you have enough stimulus, that will get the action potential to fire or get the dog to come back to you. Okay, so that's just kind of a interesting way to think about it. I kind of liked that analogy. It was very helpful, I think, in understanding the difference between these two. Okay, so now that we know that action potentials travel along axons of neurons, something you might want to consider is how fast do they do that? And the answer to that is it depends. It depends on if that axon is myelinated or if it's non-myelinated. So in non-myelinated neurons, action potentials are conducted much more slowly in this process called continuous conduction. And so in continuous conduction, the voltage-gated sodium-potassium channels are regenerating the action potential at each point along the axon. So the voltage doesn't decay. And the conduction is slow because it takes time for the ions and for the gates of the channel proteins to move. And this has to occur before that voltage can be regenerated. So non-myelinated neurons conduct their action potentials relatively slowly in the process called continuous conduction. Alternatively, myelinated axons use something called saltatory conduction. And you might have heard this in the video that I played for you guys a little bit ago. But saltatory conduction is actually like 30 times faster than continuous conduction. And the myelin sheaths, which we know are present in some of these neurons, insulate the axon and they prevent the charge from leaking out. So the voltage-gated sodium channels, they're located at the gaps that are between each myelin sheaths. And these are called the nodes of Ranvier. And the action potentials are only generated at those gaps. And so because of this, essentially what it would look like is that it's going to look like the action potential is jumping from gap to gap. And this jumping from gap to gap can occur much more quickly than moving straight along. Okay? So let's talk about why that matters, about how fast an action potential moves along the length of the axon. And we're going to talk about it in a clinical sort of way and connect it to a disease called multiple sclerosis. So a lot of times it's just called MS in shorthand. So MS is an autoimmune disease. And if you're not sure what autoimmune means, basically it means your immune system attacks your own cells because they treat them like foreign invaders. Like your immune system would think that your own cells are, you know, a bacteria or a virus or something like that. Okay, so autoimmune means your immune system is attacking your own cells. And multiple sclerosis primarily affects young adults, but what happens is that the myelin sheaths that are in the central nervous system, they become destroyed because the immune system is attacking myelin and turning myelin into these lesions called scleroses, which is where we get the sclerosis part of multiple sclerosis. And because we are breaking down myelin, that's going to slow down our impulse conduction. And eventually, impulse conduction is going to stop. Demyelinating axons increase the sodium channels. And so this is actually going to cause cycles of relapse and remission. And so some of the symptoms that you might see that a person with MS might have would be things like visual disturbances, weakness, loss of muscle control, speech disturbances, and incontinence. A lot of things that would involve sort of these voluntary actions, especially in the muscular aspect here, are going to be disturbed. And in terms of cure, there's no cure, but there is treatment, and treatment really is just going to be immunosuppressants or drugs that are going to modify the immune system's activity. And even though a person can't necessarily prevent multiple sclerosis, maintaining high blood levels of vitamin D has potentially been linked to reducing the risk of developing multiple sclerosis. So now we talked about action potentials essentially from the start all the way through the axon. And so now let's talk about what happens at the end of the action potential. What's the point of having this electrical signal be transduced down the axon? And we're going to talk about that in the sense of the synapse. So let me just do a quick little doodle here. We have I'm going to draw a neuron. Okay, so here's my neuron, and at this point, we've already talked about the action potential traveling down the length of the neuron, and eventually what's going to happen is it's going to reach this point down here. And this is going to be where the synapse is occurring. And the synapse is going to occur either so we could have a synapse occurring onto a dendrite of another neuron, or the other situation is we could have a synapse occurring onto an effector, such as maybe muscle cell. Okay. So I apologize for my barking dogs. I've apologized 100 times. You guys know my dogs bark a lot and they bark at nothing. So I apologize for that. But let's continue. So this synapse, then we're talking that it can either occur on from one neuron to the next neuron, or maybe from the neuron to an effector cell like a muscle cell. And we talked about the neuromuscular junction already. So that should kind of give you a good idea as to what I'm talking about when I'm saying the synapse. And the idea of this is that the nervous system only works because information can flow from neuron to neuron or neuron to effector. And the synapse is just this functionally connected area that mediates the transfer of information from the neuron to either another neuron or to an effector cell. Okay, so clicker. That one should only take you a second, hopefully. All right, so let's talk about the synapse then. We have a few different components, none of which should be anything too new because we did just talk about the neuromuscular junction within the last couple of weeks. So the components of the synapse are going to be the presynaptic neuron. And so this is the neuron that is going to conduct the impulse towards the synapse. And so basically it's the one that's sending the information. Okay. And then we're going to have the so here, let me just show you in this picture. This is the purple part is the presynaptic neuron. So this is the one that's sending information. And then we're going to have the axon terminal. And the axon terminal is going to be the part that's releasing neurotransmitters into the space called the synaptic cleft. And the synaptic cleft or the gap. It's going to be between the neurons or between the neuron and an effector that's going to prevent that neuron to neuron electrical transmission, or neuron to effector electrical transmission. And then you're going to have a postsynaptic neuron, which is going to be either the dendrite or a cell body that's going to be receiving the impulse. So let's talk about these synaptic connections. There's a few different types that there could be, and that's why I'm going to bring up the synaptic connections here. So we can have axodendritic connections, which will occur, see it over here, between the axon terminals of one neuron. So this would be the presynaptic neuron, and the dendrite of another neuron. Okay, so here's a dendrite of a different neuron. Okay, and so this would be the axodendritic, from axon terminal to dendrite. Or the other type would be axosomatic. And axosomatic synaptic connections occur between the axon terminals of one neuron, the presynaptic neuron, to the cell body of another neuron. Okay, and so that would be the somatic part, refers to soma, and soma means cell body. Some less common connections, though, that might happen, you might have axoaxonal connections. So those would go from, say, if it was this axon terminal and it directly connected to this orange axon terminal, that would be axoaxonal. Dendrodendritic, this is going to be a connection from, let's pretend that there is, okay, let's pretend that this dendrite comes out here and decides to touch that one, right? And there's some kind of connection right there. That's going to be dendrite to dendrite, or dendrodendritic. Try saying that fast, not easy. Alternatively, you might have a connection that's going to go from the soma of one cell to the dendrite of another cell, and that can happen as well, but those are much less common connections. So not only do we have different pieces that can potentially come together, synapses themselves are usually, or they're one of two types, okay? So they're either electrical or they're chemical. In terms of electrical synapses, these are very uncommon. And so here, in electrical synapses, the synaptic cleft is kind of like a doorway, and messages move directly from one room to another room. And so in adults, electrical synapses are found in regions of the brain that are responsible for normal stereotyped movements, like the normal jerky eye movements that we do. And they can also, these types of synapses also occur in the hippocampus of the brain, which is the part of the brain that's responsible for learning and for memory. But in general, most electrical synapses occur during embryonic development before the nervous system's fully developed. The more common type are the chemical synapses. And chemical synapses rely on the release, the diffusion, and the reception of neurotransmitters. And so chemical synapses are going to include the axon terminals of presynaptic neurons, a synaptic cleft, and then a receptor region on a postsynaptic neuron. But you guys should be relatively familiar with this type because the neuromuscular junction is an example of a chemical synapse. But in case you don't remember, let's watch a quick video.
There is a narrow gap, the synaptic cleft, between the synaptic terminal of a sending neuron and the surface of a receiving cell. An action potential arrives at the end of the axon of the sending cell. This causes chemical changes that make vesicles containing a chemical called a neurotransmitter fuse with the plasma membrane. The neurotransmitter molecules spill out into the synaptic cleft, diffuse across, and bind to the receptors on the receiving cell's membrane. The binding of neurotransmitters to receptors causes the attached ion channels to open. Depending on the type of neurotransmitter, receptors, and channels involved, this may excite or inhibit the receiving cell. Here we show excitation. Sodium ions rush in, changing the polarity of the membrane, and an action potential is triggered in the receiving cell. The neurotransmitter is broken down by an enzyme, and the ion channels close. Enzymatic breakdown is one of the several mechanisms by which transmission is terminated at chemical synapses.
Okay, so just going to go over this more step-by-step sort of situation here. Just to refresh your mind in case you're having a hard time recalling this chemical synapse thing. So chemical synapse transmission occurs in 6 steps. So step 1, action potential is going to arrive at the axon terminal. Okay, so here we have a presynaptic neuron. We have a synaptic cleft, which is this hollow space. And then we have a postsynaptic neuron. So we're going to say this neuron, let me just to make it a little bit sort of easier to grasp, I'm going to do this. Presynaptic neuron. So this is neuron number 1, outlined in red. And then we're going to have neuron number 2 is sort of this purple colored one that's that I just outlined in purple. Okay, so neuron number 1 is going to have an action potential arrive at its axon terminal. When that action potential arrives, voltage-gated calcium channels open, right? Because an action potential is an electrical signal. And so that's going to change the membrane potential that's located here. And that membrane change, it's going to be a voltage change, is going to cause these calcium channels to open. And this is going to lead to calcium entering the axon terminal. Okay. Step 3, calcium entry is actually going to cause these synaptic vesicles to release their neurotransmitters via exocytosis. So these vesicles, they're going to fuse with the membrane here at the end of the terminal and spill out their contents into the synaptic cleft. The spilled out contents are called the neurotransmitters, and the neurotransmitter is going to diffuse across the synaptic cleft, and it's going to bind to a specific receptor that's going to be located on the membrane, the receiving membrane of neuron number 2, okay? Also called the postsynaptic membrane. So if we take a look at one of these, excuse me, one of these channels here, and we just zoom in on it, the next thing that's going to happen is the binding of the neurotransmitter is going to open this ion channel, and that's going to create a graded potential. Okay, so this one, this particular ion channel, if it relies on the binding of a neurotransmitter, you guys should be thinking, okay, this is a ligand-gated ion channel or chemical-gated ion channel. Okay, so the binding of this neurotransmitter opens the ion channel and creates a graded potential. After this happens, the neurotransmitter, after a few milliseconds, the effects of the neurotransmitter is going to end. And it's going to end by either the neurotransmitter through reuptake. So the neurotransmitter is going to unbind from its receptor on the postsynaptic neuron and get taken back up into the presynaptic neuron. The other thing that can happen is that the neurotransmitter can be degraded or broken down, basically, into its components. And then the third thing that can happen is that the neurotransmitter, once it is released back into the synaptic cleft, it might just diffuse out here into space and get lost. Okay, diffused away from the synapse. So I'm just going to take a second to explain this one. If you guys remember the calcium channels from 2 slides ago, go back 1 second, right? So these calcium channels, if you block the calcium channel, what can't happen? If calcium can no longer enter the cell, then these vesicles can no longer fuse with the terminal, and the neurotransmitters can't be released. And so that's why A is the correct answer. If you expose a neuron to a calcium channel blocker, then the neurotransmitters can't be released from that presynaptic neuron. So I'm going to again briefly explain this one just because it's more of a thinking beyond type of question. So there's certain drugs that when taken can keep neurotransmitters in that synaptic space, in the synaptic cleft, longer than normal. And so the way that these drugs work, they prevent the neurotransmitter from being broken down by enzymes. By either they can directly inhibit the enzymes that are supposed to break down the neurotransmitter, or they can basically block astrocytes or the presynaptic terminal so that the neurotransmitter can't be brought back up into the presynaptic terminal. And so an example of a psychosomatic drug that would do this sort of situation are called SSRIs, or selective serotonin reuptake inhibitors. And so if you're not sure what those are, I encourage you to look at the pharmacology of SSRIs, or selective serotonin reuptake inhibitors. It's really quite interesting once you understand how the neurotransmitters are working, okay? So that leads us to our last part of the lecture for today, which is all about neurotransmitters. And so by now, we kind of have an understanding that the neurotransmitters are released, they bind to postsynaptic neurons, and then something happens. All right, and so basically the neurotransmitters are kind of like the language of the nervous system. It's how the neurons are talking to each other. And actually more than 50 neurotransmitters have been identified, and most neurons make 2 or more different types of neurotransmitters. And this is so neurons can exert several different influences. All right, so think of your neurons as being like bilingual or trilingual. All right, so they can speak more than one language so that they can talk to everybody in the whole population. Usually, different neurotransmitters are released by different stimulation frequencies. So basically what that means is that if you, I don't know, stimulate a neuron so many times a certain way, then it's going to release neurotransmitter A. But if you stimulate the neuron at a different frequency, that it's going to release the different neurotransmitter. Okay, and so neurotransmitters, they're classified based on their chemical structure and also on their function. So in terms of chemical structure, we'll go through the classes here. And so first and foremost, acetylcholine is kind of in a class of its own because it was the first identified neurotransmitter and is the best understood one. So if you guys remember back to muscle tissue lecture, acetylcholine is released at the neuromuscular junction. But in addition to being present there at the neuromuscular junction, acetylcholine is also used by many autonomic nervous system and some central nervous system neurons. And so acetylcholine is actually made from acetic acid and choline. And the enzyme that makes acetylcholine is called choline acetyltransferase. And acetylcholine gets broken down or degraded by an enzyme called acetylcholinesterase. Okay, so acetylcholinesterase is going to break acetylcholine down. So I'm gonna just write out a quick little tidbit here just to make what I'm saying seem easier to understand. Okay, so if you have acetic acid plus choline you're going to get acetylcholine, but this can't happen unless it happens by choline acetyltransferase. Okay, so in this direction happens thanks to acetylcholine transferase. Kind of moving the opposite way, if we want to break acetylcholine back down into its two original components, that's when we use acetylcholine esterase. Okay, that breaks acetylcholine back down into acetic acid and choline. Right? So moving on. The next chemical class here of neurotransmitters are called amines. Sometimes they're called biogenic amines, sometimes they're called biologic amines. But these amines, all of them are widely used in the brain, and they play roles in things like emotional behavior and the biologic clock. Sometimes they're used by autonomic nervous system neurons, specifically the autonomic motor neurons. Especially norepinephrine is used. And imbalances in these biologic amines are often associated with mental illness. And so our biologic amines include our catecholamines, which includes dopamine, norepinephrine, and epinephrine. And then our indolamines, and that includes serotonin and histamine. Okay, so all of these are biologic amines. They're all neurotransmitters. The next chemical class is our amino acids. And so this includes GABA, which is gamma aminobutyric acid. So that's what GABA is short for, gamma aminobutyric acid. And something interesting about GABA is that it's augmented by alcohol and anti-anxiety drugs. Another amino acid is glutamate, which is an excitatory neurotransmitter, meaning it's going to promote depolarization of a neuron. And so glutamate is really important for learning and for memory, but it's also known as the stroke neurotransmitter. So excess amounts of glutamate actually can cause neuron death by ischemia, which is oxygen deprivation that's usually caused by a blood vessel being blocked. The other type of amino acid neurotransmitter would be glycine, and glycine is inhibitory in the spinal cord. And so basically, inhibitory neurotransmitters prevent action potentials from being fired and relayed. So this like blocks signals. And if we block the blocker, then that can result in things like uncontrolled convulsions and respiratory arrest. So this is a drug that's going to block glycine. And if you block a blocker, that's going to lead to basically free flow, right? And so this would be the example here. So uncontrolled convulsions. Normally you're not convulsing. Normally you're just sitting there chilling, right? But if you take away, if you block the neurotransmitter that's keeping you in that state, then that's when you can start convulsing. The next chemical class of neurotransmitters are peptides. Specifically, usually they're called, in the context, they're called neuropeptides. And these are just strings of amino acids. And so these are things like endorphins, which are basically these natural opiates that inhibit pain and mimic heroin and morphine and methadone. We also have tachykinins, which mediate pain transmission in the peripheral nervous system. And in the central nervous system, they're involved in respiratory and cardiovascular control, as well as the mood of a person. The next type of peptide on this list is somatostatin, and somatostatin is often released at the same time as GABA, and a couple of the effects that somatostatin has would be that somatostatin inhibits growth hormone release, and it also plays a role in regulating digestion. And then finally, cholecystokinin, or CCK, is often involved in anxiety, pain, and memory. But most importantly, CCK is the gut-brain peptide hormone that inhibits appetite. So this next chemical class, they're called gasotransmitters and lipid transmitters. It's a super interesting class because it brings about the idea that something as simple as gases can cause communicating signals between neurons. It's kind of incredible. But an example here would be nitrous oxide, which the release of nitrous oxide potentiates stroke damage. And the release of nitric oxide is also involved with enhancing some types of impotence. So basically, this is how Viagra works. Carbon monoxide is another gasotransmitter and has been found to affect the neuromuscular and neuroglandular synapses. Specifically, in this case, it can cause suffocation. Hydrogen sulfide is the most recently discovered gasotransmitter, and we're not 100% sure on all of its effects, but we do know that it somehow alters the functions of proteins in the body. The lipid transmitters in this class would be things like endocannabinoids, which are involved in memory, appetite control, nausea, vomiting, and also neuronal development. And the receptors of endocannabinoid receptors are actually activated by THC that's in cannabis. Finally, the last chemical class here are purines. And so with this chemical class, we have things like ATP. And before I talk about that, I just want to say purines are the monomers of the nucleic acids, and they have effects both in the central and the peripheral nervous system. And so ATP, which we know is our energy molecule, is actually now considered a neurotransmitter. And it's released by sensory neurons to provoke the sensation of pain. And the other molecule here is adenosine. And adenosine is a very potent inhibitor in the brain. And so adenosine is responsible for our sleep-wake cycle and also for terminating any seizures. It works by dilating the arterioles and increasing blood flow to the heart and other tissues as needed. And an interesting thing about adenosine is that it's actually blocked by caffeine. So caffeine is a stimulant that's going to block the receptors for adenosine in the brain. And because adenosine is responsible for maintaining our sleep-wake cycle, too much caffeine can disrupt that sleep-wake cycle. So the last couple of slides here just have to do with the functional classification of neurotransmitters. And so if we're grouping neurotransmitters based on their function, we can group them based into two classifications. So we can group them based on their effects, or we can group them based on their actions. So in terms of their effects, neurotransmitters can either be excitatory or inhibitory. And excitatory neurotransmitters cause depolarization, whereas inhibitory neurotransmitters can lead to hyperpolarization. And the effect that a neurotransmitter has is just dependent on which receptor it binds to. And so I'm just going to take a second to talk about that. So we're going to say that we have two receptors here. And if our neurotransmitter comes in and it binds to this receptor, then we're going to say that it's going to have an excitatory effect. Okay, so if the neurotransmitter binds to this one, then it causes excitation. However, if that same neurotransmitter instead binds to this receptor, then what can happen is that it can have an inhibitory effect or a hyperpolarizing effect. So it really just depends on which receptor the neurotransmitter binds to, because it might have multiple receptors. An example here, just to kind of go on this effect mantra here, is GABA and glycine are usually inhibitory neurotransmitters. So they only have these inhibitory receptors. Glutamate only has a excitatory receptor. And then we have acetylcholine and norepinephrine, which bind to at least two receptor types with opposite effects. And so this kind of stamps back to the example that I was kind of giving you guys earlier, that say this is acetylcholine. This is also acetylcholine. If it binds to this receptor, then so this receptor we're going to say is at the neuromuscular junction. If acetylcholine binds to that receptor, it's going to cause an excitatory response. But say we're in the heart and acetylcholine binds to the receptor that's there for it, which actually happens to be an inhibitory receptor. Okay, so if acetylcholine binds to the receptor that's located in the heart, then that's an inhibitory receptor, and that's going to cause things like a slowed heart rate. Okay. So the other way that we can classify these neurotransmitters based on their functions are by their actions. And specifically whether or not they are have a direct action or an indirect action. So if we're talking about a direct action, that's when the neurotransmitter is going to bind directly to and open an ion channel. And this promotes a really rapid response by altering the membrane potential. So an example here would be like acetylcholine at the neuromuscular junction. When acetylcholine binds, it opens up that ligand-gated ion channel, causes a membrane potential to change. You have a fast response, right? Alternatively, some neurotransmitters don't work like that. Some neurotransmitters have an indirect action, and so this is when a neurotransmitter is going to act through second messengers, and usually these are G protein couple receptor pathways. And basically, in this sort of scenario, the neurotransmitter is going to bind to a receptor, which causes another change, which causes another change, which causes another change, which leads to an overall change inside of the cell. So these are indirect actions are much broader, longer lasting effects. It's kind of similar to hormones, which we'll get to talking about at the beginning of AP2. So example here of neurotransmitters that act indirectly are our biologic amines, our neuropeptides, and our dissolved gases. So in conclusion, all of the events that we discuss in this section lead to the communication between all of the neurons that are in our body. Remember that this section, we discuss things that are occurring on really a microscopic level. But in the next unit, we're going to be covering the central nervous system and then the peripheral nervous system. We're going to talk about how all of the axons and all of these neurons work together and use action potentials to carry out whole body functions. Okay, so that is all I have for you guys for this week. I hope you have a great weekend. Stay safe, stay healthy, and I will talk to you guys next week.